LemnaTec Academy, an introduction to phenotyping for biological applications. Phenotyping of plants, seeds and non-plant organisms. LemnaTec is the global leader in plant phenotyping, established in 1998. We offer phenotyping systems for laboratories, greenhouses or growth rooms, and also for outdoor applications. All systems include data analysis, data interpretation, and value generation. Since 2019, LemnaTec is part of the Nynomic group. The group consists of 10 companies, all active in optical sensing for various applications. LemnaTec has customers all over the world. The customers are research institutes, universities, and companies active in research and breeding. What are phenotypes and how can phenotypes be measured? The phenome is the sum of all phenotypes of an organism. Phenotypes are the outer properties. They are influenced by the physiology, and the physiology is controlled by the genome. In case of plants, also the environment takes a major influence on the physiology and therefore also on the phenotypes. The phenotypes are a result of the interaction between the genetics and the environment. It can be considered like an equation where genes, gene expressions, also enzyme activity or metabolic activity interact with environmental factors like light, soil, air, weather conditions, or stress factors to result in phenotypic properties. Such properties are dimensions, morphology, and color of, for instance, leaves, shoot, root, fruits, flowers, and also the overall biomass, the water content, or the abundance of pigments. An important feature is also the photosynthesis. So now, what is phenotyping? It is measuring and quantifying phenotypes. It is linking the phenotypes to gene functions and also linking the phenotypes to environmental factors. There are many factors that drive phenotypes. The genetics, in particular in case of plant breeding, technologies that modify the genetics of crops. But on the other side, there are also a lot of environmental factors like weather conditions, pathogens, pests, and human-derived stress factors like pollution and the climate change. Well, that's the theory. In practice, phenotyping is also about sample preparation, the measuring process, and about the measuring technology, because all these factors also define how the phenotypic outcome of a measurement is. The phenotypes that are measured depend on the way how the measurement takes place and how the samples are presented for the measurement. Measuring phenotypes is not a very new activity. It has been taking place more or less since humans started working with plants, and also the famous geneticist Gregor Mendel has been working on phenotypic measurements as he was classifying the pea plants where he made his genetic studies according to shape and color of the fruits of those plants. Complexity increases in the different levels of the analysis in plant physiology, while genomic analysis takes place on the level of DNA, transcripts, proteins and metabolites already cope with a multitude of substances, and the phenome as a result of all these physiological activities is again more complex. Working with phenotypes is about descriptions, measurements and interpretations. On top there is the phenotype of the organism as such. In a human inspection we see and describe what is present, and as next level we use imaging technology to take photographs, scans and spectra of the visible parts of the phenotypes. This is followed by software and computing that processes and analyzes images and data. There can be machine learning and artificial intelligence for interpreting the features, and in the end it is possible to set up mathematical models for growth, development, health and more features of the phenotypic properties of the organism. LemnaTec phenotyping hardware software has the task of image acquisition, image processing and analyzing, and, if required, machine learning. How does it work? How are imaging technologies used in phenotyping? Images describe phenotypes. On the photo, you see two plants having totally different phenotypes. They differ in size, shape and color. What the image does not tell is why the phenotypes are so different. Phenotyping, measuring phenotypes, is an important tool to explain differences in phenotypes. Translating images into numerical data. Having images, a phenotyping software is required to process the images and in the end to convert image data into numerical data that can be interpreted for the biological properties of the sample. The typical basic steps in image processing are defining a region of interest, converting the images, setting masks, and defining objects that are listed in the end. Such an object list can then be used for counting, determining areas, shapes or color information. These steps essentially work for many plant species, for many varieties that are of interest in research and breeding. What do phenotypes tell about the plant? Let's have a closer look with an example. Here you see a tray with different plants. All plants are Arabidopsis thaliana. All have been grown with the same environmental conditions. They differ because they are different ecotypes, so they differ on the level of genetics. They do not differ on the level of environment because the conditions have been kept same for all the plants. The other way around, no difference in genetics, but difference in environment. Again, Arabidopsis plants, they look different, but in this case it is about having differences in water availability, moist soil or wet soil with 10% more water, or bright light and low light. The combination of moist soil and bright light, but also the combination of wet soil and low light, results in equal sizes of plants at the end of the experiment. However, the dynamics, how they grow to that final size, is different in both cases. The plants having low light from the beginning start growing slow and speed up the more leaf area they grow. The other plants having bright light are growing fast from the beginning, even at low leaf area, but then they slow down growth because the soil moisture becomes limiting. The plants with moist soil and bright light grow most compact throughout the whole time of growth. The plants at low light are not compact because they develop long petioles to reach out of the darkness, so they avoid the shade. This example shows that phenotypic data in combination with background information about the plant growth conditions can lead to explanations of plant growth behavior. It shows also that dynamic data in time-resolved repeated data acquisitions delivers additional data that are not available in one-point acquisition at the end of the experiment. So how to come from research to breeding? Precision plant phenotyping can serve to derive a model of plant growth that explains how plants will behave later, even if you only have measurements on young plants. The capability to phenotype a whole crop plant life cycle from germination to maturation of the seeds is even more valuable because you can monitor the whole development throughout the life cycle of the plant. A typical example for the pathway from images to data is taking a measurement, deriving parameters and information, and then generating knowledge out of those measurement data. In practice, that means taking images, measuring the plant area data from the images, using those area data for biomass calculation, and comparing biomasses across genotypes and treatments for QTL discovery. Having spoken already about taking images, what are the typical imaging principles and their application cases in phenotyping? This table gives an overview on the imaging technologies, the measured parameters, and the derived biological information. The image acquisition and deriving parameters is what the phenotyping system does. The biological information is what you can do with the output of a phenotyping system. The table makes the overview. The next pages will give the details on the different imaging principles. Processing visible light images. It is possible to count things, to measure sizes, describe shapes, and analyze colors. Derived biological information can be growth, biomass, development, stress, or activity of the samples. Chlorophyll fluorescence imaging is the dynamic measurement of chlorophyll fluorescence after exciting the fluorescence with specialized illumination. It gives information about the status and activity of the photosystem too. It is possible to derive photosynthesis-related parameters that in turn are indicators on physiological processes, stress, or pathogen responses. The setup consists of LEDs for fluorescence excitation and a camera that records fluorescence signals. In multispectral imaging, images are taken in a range of distinct wavelengths. These images can be used for calculating vegetation indices or carrying out analysis, for instance, corresponding to pigmentation. In hyperspectral imaging, images are scanned along broad ranges of wavelengths, for instance in the V-near or in the S-sphere range. These hyperspectral data cubes not only can serve to calculate vegetation indices, but they also provide spectral signatures, for instance of pigmentation, water status, stress or disease. In near-infrared imaging, a filter is used at the 1450 nanometer water band. These images serve to assess the water content of the samples. Infrared images can be used to assess the surface temperature of samples. The surface heat emission from samples is, for instance, indicative of transpiration and can be used as a stress indicator. In bioluminescence imaging, luminescence is detected with a high-sensitivity camera. Luminescence functions are, for instance, used for labeling microbes or gene products and metabolites. The luminescence images can be overlaid with visible light images to combine luminescence signals with phenotypic properties. Laser scanners provide point clouds corresponding to the sample surfaces. They can be used to calculate dimensions and morphology of the samples. All recorded images and scans need to be processed with phenotyping software to derive phenotypic properties out of the imaging data. The core function of such a software is programming and image processing. With LemnaGrid, LemnaTec provides a platform for graphical programming of image processing workflows. There are functions in a library that can be combined, and each block out of the library has a function in image processing. Typical functions are segmenting. This means separating the plants from the background. When something is separated from the background and recognized as an object, it can be measured. In this example, small seedlings are detected as they emerge out of a multi-pot tray, with some pots having already a seedling and others do not yet have a seedling. The result of the analysis is the number of emerged seedlings and the size of each individual seedling, together with color and morphological information on the seedlings. For complex analysis, we can use machine learning. This process consists of two parts. The first part is the training, where the user determines what to look for. To do this, images are labeled and a prediction model is set up. Once the model is established and working, it can be applied. The trained model does a routine job. For instance, it classifies many images of a certain type and puts out results according to the previously defined criteria. The main work for the user starts once the parameters are put out by the phenotyping system, because this means the machine has done the job and now it's up to the user to interpret the data in the context of the biological questions that underlie the study. For more information, please contact me at LemnaTec according to the given contact data.